Hello, hello. This is Milo. I will be your host for this beautiful afternoon. So, as many of you know, I'm a layman. I don't know much biochemistry, but I brought in 3 biochemical experts to help me figure out what these protease things are. I can't put it together. I'm worried about the final, but I have hope. I have hope that they will help me out here. So I'm going to give the mic over to Rashawn. Hi, Rashawn. How are you?
Hi, I'm good. Thank you.
Good.
So the first thing, just defining proteases.
Yes.
Proteases are enzyme that breaks down proteins into amino acids, and their function is cleaving peptide bond with water, and they are part of hydrolase class of enzyme.
Mm-hmm.
And they have several components, actually 3 components. One of them is catalytic residues, and in the active site helps forming nucleophile. Second one is oxyanion hole, and oxyanion hole is a structural component that stabilizes intermediate tetrahedral intermediate and by providing space.
Okay.
The third one is recognition site, substrate specificity pockets. And the last one, the last thing that I wanted to go over is the cis bond. It's the bond where the enzyme cuts, and it cuts to the right side of the cis bond, which is P1 prime, and that's where that side is what binds to the SI pockets.
Uh-huh. Okay, well that was helpful, thank you. And we're gonna move on to James here. James, I know you're gonna talk about serine proteases, something like that.
Yeah, serine proteases, because they're really important in biology. Because
are they though?
Well, I mean, if you want your blood to clot when you're cut,
I do. I would like that.
I think you would want some proteases around. So what serine proteases do, and this actually involves cysteine proteases as well, but we're just going to call them serine proteases.
Sure.
And the reason why they have this name is because serine and cysteine are the residues in the active site that actually participate in the nucleophilic reaction.
And it's just them?
No, there's actually 3. So going back to what Rashawn said about there being catalytic residues, there's a catalytic triad. And so in serine proteases, you have serine, you have histidine, and you also have aspartic acid. And so when the substrate comes in, the electron pair on the oxygen of the serine molecule is going to attack the carbonyl carbon of your peptide bond, and that's what's going to initiate the nucleophilic reaction. And then you have histidine, which can act as both a general acid or a general base at physiological pH. And because it's able to do that, it's able to help move the mechanism along. And so serine proteases, again, they're really important for all biological functions.
Okay, great. Yeah, thank you so much. That's helping. I'm starting to get a picture of what these protease things are. I don't know. So we went over serine proteases. We went over the cysteine proteases. We did a general discussion about what proteases are, the type of enzymes that they belong to. Is that it? I don't know. I have another expert in here. This is Joe. Joe, you can leave your lab coat at the door. Thank you. Are there any other type of proteases?
Hello, Milo. Thank you for having me here, first of all. Thank you for the lovely introduction. And yes, there's 2 more types of proteases that I would like to discuss.
Love to hear.
They're not as big of players as, say, serine or cysteine, but I think they're still pretty cool.
Okay.
And they're called aspartyl and metallo. And that they're both pretty similar to each other in that they both use water as a nucleophile. They basically use water to attack the peptide bond because they both have a group that removes a proton from water, and the negative OH is the nucleophile that then attacks the bond at carbon. So that's pretty much it for aspartyl and metallo, but I would also like to add about another reason why we care about proteases at all is that, they're very important to our bodies in how we handle our DNA, basically, and how we replicate and transcribe DNA. And
so the proteases play an important role in transcription.
Oh yeah, definitely, man.
Okay.
So I would say that they're very important, and aspartyl and metallo are also. So that's it.
Okay, that sounds great. You heard it here first, folks. Proteases, it's gonna be big. Thank you.